Bismillahirrahmanirrahim. Assalamu alaikum students. We continue with our discussion of immunity. Students, today we will be discussing cell mediated immunity. Cell mediated immunity. Students, by definition, cell mediated immunity is defined as a type of acquired or adoptive immunity which involves different types of immune cells. What are those different types of cells? Number one, macrophages. Number two, T lymphocytes. And number third, natural killer cells. Students, these are three main cellular components of cell mediated immunity. I repeat, number one, macrophages. Number two, T lymphocytes. And number third, natural killer cells. Okay students, we discuss these cells one by one. Now, first of all, we move with macrophages. Students, as you all know, that the primary function of macrophages is phagocytosis. What we mean by phagocytosis? In very simple words, phagocytosis means engulfment of foreign antigen or microorganisms which enters the host. Okay, the primary function of macrophages is phagocytosis. Number two, antigen presentation. Students, this function of macrophages, antigen presentation, is the hallmark of cell mediated immunity. What is antigen presentation? We will discuss it later on. Now students, the third function of macrophages, number one, phagocytosis, number two, antigen presentation, and number third, production of cytokines. Production of cytokines. These cytokines, they involve different types of interleukins which are produced by the macrophages, and in turn these interleukins, they activate the other cellular components of cell mediated immunity. This was brief about macrophages. Now students, we move on to T lymphocytes. Students, T lymphocytes, these are the main cellular components of cell mediated immunity. There are two subtypes of T lymphocytes. Number one, Cd8 helper T cells, and number two, Cd8 cytotoxic T cells. Students, these both are subpopulations of T lymphocytes. Now, to elaborate further, Cd8 helper T cells, they are again divided into two main subtypes: thymic 1 cells and number two, thymic 2 cells. CD4 helper T cells, they have two further subpopulations, thymic 1 cells and thymic 2 cells. Now students, what are CD4 and Cd8 cytotoxic T cells? To be very simpler, I will tell you that CD4 helper T cells are those types of T lymphocytes which have CD4 surface proteins on their surface. Students, in very simple words, CD4 helper T cells are those T lymphocytes which possess CD4 surface proteins on their cell surface. And students, these CD4 helper T cells, they recognize those antigens which are in combination with class 2 MHC proteins. I repeat, CD4 helper T cells recognize those antigens which are in combination with MHC class 2 proteins on the surface of antigen presenting cells. Now, Cd8 cytotoxic cells. Students, Cd8 cytotoxic T cells are those T lymphocytes which possess Cd8 surface proteins on their cell surface. And these Cd8 cytotoxic T cells, they recognize those antigens which are in combination with class 1 MHC proteins on the surface of antigen presenting cells. Clear? Now students, the process of cell mediated immunity starts when entering microorganisms which enters the host, they are engulfed either by macrophages or they are taken up by the dendritic cells. Now these invading microorganisms, they are broken down into smaller peptides. And these peptides, they are then presented on the surface of antigen presenting cells in combination with class MHC proteins. Okay? In simpler words, I repeat that the invading microorganism is broken down into peptides in either of macrophages or dendritic cells, or you can say antigen presenting cells. Now these peptides in the form of antigenic material, they are expressed on the surface of antigen presenting cell in combination with MHC proteins. Students, at this level, I will tell you a very simple concept that for practical purposes, MHC proteins or HLA antigens, both terms are same. MHC proteins, these are genetic molecules located in HLA antigens. And these are located on the long arm of chromosome 6. Okay? Coming back to our point. Now, once they antigen has been presented on the surface of antigen presented cell in combination with MHC proteins. Now these antigenic material, they are produced in front of T lymphocytes, and later T lymphocytes becomes activated and carry out their next functions. After becoming activated, T lymphocytes again they produce different type of cytokines. These may be different types of interleukins, and on the basis of these interleukin productions, we can divide the function of T lymphocytes into two components, students. Number one, effector functions. Students, effector functions of T lymphocytes is in the form of two functions. Number one, delayed hypersensitivity. Delayed hypersensitivity. Students, delayed hypersensitivity mechanism is carried out through interleukins through cytokines like gamma interferons, macrophage activation factor, and number third, macrophage migration inhibitory factor. These four cytokines are involved in the mechanism of delayed hypersensitivity, and this delayed hypersensitivity involves macrophages and T lymphocytes. How it involves? It mainly involves through production of gamma interferons, macrophage activation factors, macrophage migration inhibitory factors. This function of effector function of T lymphocytes, delayed hypersensitivity. Now, number two effector function of T lymphocytes is cytotoxicity. Students, cytotoxicity is mainly seen in virus infected cells, graft rejection cases, tumor cells, and it is mainly carried out by Cd8 cytotoxic T cells. Cd8 cytotoxic T cells, they produce perforins. Cd8 cytotoxic T cells, they produce perforins. and it is because of these perforins that the infected cells are killed through the process of cytotoxicity. Number second component or released by CD8 cells is granzymes. Granzymes. Students, granzymes are proteases, again, which helps in cytotoxicity. So we can say that the main effector functions of T lymphocytes are two. Number one, delayed type hypersensitivity, and number two, cytotoxicity. Now students, the second component functions of T lymphocytes is regulatory functions. The regulatory functions of T lymphocytes include number one, antibody production. Students, CD4 helper T cells, they activate beta cells to transform into plasma cells, which then produce antibodies. Okay? Antibody production, the main regulatory function of T lymphocytes. This is carried out through CD4 cells. Number two, cell-mediated immunity. Students, it is regulatory function of T lymphocytes. Cell-mediated immunity. And number third, suppression of certain immune responses. Suppression of certain immune responses. Students, this word means that different immunogenic responses are decreased or diminished by T lymphocytes. Example is lepromatous leprosy. Students, in lepromatous leprosy, delayed hypersensitivity mechanism is suppressed. The patient shows no response when checked with the antigen. There is no type 4 hypersensitivity or delayed hypersensitivity reaction in such patients. So I conclude that the function of T lymphocytes are mainly of two components: effector functions and regulatory functions. Effector functions, I have told you, there were two main effector functions of T lymphocytes. Number one, delayed type hypersensitivity, expression of delayed type hypersensitivity, and number two, expression of cytotoxicity. Both of these mechanisms, they are actually the indication of activation of T lymphocytes. Now, what are the regulatory function of T lymphocytes? Students, the regulatory functions include number one, antibodies production. Number two, cell-mediated immunity. And number third, suppression of certain immune responses. Okay? I conclude with some of the important points of cell-mediated immunity. Number one, students, cell-mediated immunity is that type of acquired immunity which involves cellular components. And these cellular components, they are mainly macrophages, T lymphocytes and dendritic cells. Okay. Number 2 characteristic feature of cell-mediated immunity is cell-mediated immunity is mainly effective against intracellular bacteria, virus-infected cells, tumor cells and allograft. Okay, students? Third characteristic feature of cell-mediated immunity, that cell-mediated immunity involves T lymphocytes and antigen-presenting cells. Students, there are 3 common types of antigen-presenting cells. We commonly call them as APCs. What are antigen-presenting cells? Macrophages. Number 2, natural killer cells. Number 3, B lymphocytes. These are main antigen presenting cells. And these antigen presenting cells, they present the offending antigen towards T lymphocytes, which are later activated, and the 2 subpopulations of T lymphocytes, that is CD4 helper T cells, Cd8 cytotoxic T cells, they become activated, and later on the cell mediated immunity function is performed. Students, a very small point at this level is how to measure the immune status of the function. In very simple words, if the patient is having effective delayed type hypersensitivity or type 4 hypersensitivity reaction, it means that the patient is having effective cell-mediated immunity in his or her body. In conditions like HIV or AIDS, this cell-mediated immunity, it is compromised, it is suppressed, and patient shows no delayed hypersensitivity response to the offending antigens. So it was all about cell-mediated immunity mechanism. In our next lecture, inshallah, we will be discussing antibody-mediated immunity. Thank you, students.